Most people think spider silk is just sticky thread. Then you watch someone match a collected strand against an equal length of steel wire, and the silk wins. That should be the end of the surprise. It isn't. The deeper you go into how a spider actually works, the more the ordinary picture fails. The material is only the first lie. Picture the demo. Someone has collected dragline silk directly from a spider's spinnerets, the structural thread, not the glue-coated capture spiral, but the thick outer frame thread the animal uses as its lifeline. They've matched it, strand for strand, against a wire of the same length cut from mild steel. Both are placed under load. The steel wire, predictably denser, predictably grey, sits in one jaw of the test apparatus. The silk, almost invisible at 3 to 5 microns in diameter, sits in the other. The load increases. The steel wire reaches its limit and fails. The silk is still holding. The steel comparison survives as a piece of popular lore because the underlying numbers are genuinely strange. Dragline silk, the structural thread that forms the web's outer frame and serves as the spider's safety line, has a tensile strength of roughly 1.0 to 1.6 gigapascals. That places it in the same territory as high-grade alloy steel. So far, that sounds like a draw. But silk is approximately 1/6 the density of steel. 1/6. Steel runs around 7.8 grams per cubic centimeter. Silk sits around 1.3. When you compare them by weight rather than by volume, which is what the amateur demos do, which is why the demos look the way they do, silk carries roughly 4 to 5 times the tensile load per gram. That gap follows directly from the physics of specific strength. A given mass of silk made into a thicker strand simply outperforms the same mass of steel wire. The material that does this is built from proteins called spidroins, specifically major ampulate spidroins 1 and 2, known as MASP1 and MASP2. MASP1 is primarily associated with dense crystalline beta sheet regions, stiff, tightly hydrogen-bonded nanocrystals that contribute to the thread's raw tensile strength. MASP2 is more associated with the amorphous, glycine-rich sections that contribute elasticity and the capacity to deform before breaking, though both proteins contribute to both properties through different structural motif ratios. Together, they produce a fiber that combines roughly 1.1 gigapascals of strength with the ability to stretch by about 27% before failure. High tensile steel achieves greater peak strength, but fails after less than 1% extension. Spider silk, because it stretches rather than shatters, absorbs roughly 30 times more energy per gram before breaking, a property engineers call toughness, and the number that most clearly separates silk from nearly everything else we know how to make. There's a spider from Madagascar called Chirostris darwini, Darwin's bark spider, that builds the largest known orb webs in the world, spanning rivers and streams on bridge lines, sometimes exceeding 25 meters. Its dragline silk measures a tensile strength of approximately 1.6 gigapascals and a toughness of around 350 megajoules per cubic meter, making it more than twice as tough as any other spider silk previously measured, and reportedly more than 10 times tougher than Kevlar on a volume-to-volume comparison. The female spider producing it weighs about half a gram. A cable of that silk, 1 millimeter in diameter, could support the weight of an adult lioness. This isn't metaphor. This follows from the tensile strength and the cross-sectional area by standard mechanical calculation. The silk discussed in steel comparisons is specifically dragline silk. Capture spiral silk, the sticky corkscrew threads that actually snare prey, is built differently, optimized for adhesion and extensibility rather than peak tensile strength. Viscid capture spiral threads can stretch to roughly 270% of their original length, which gives them comparable toughness to dragline silk through a completely different route. Orb weavers alone produce up to 7 distinct silk types from separate abdominal glands, each calibrated for a different mechanical purpose. Dragline for the frame and lifeline, flagelliform for the capture core, aggregate glue coating the capture spiral, aciniform for wrapping prey, tubuliform for egg sacs, and so on. The one that beats steel by weight is the structural thread. It's only the beginning of what this animal is. Before the body plan makes sense, one assumption needs to die. Spiders aren't insects. The two groups share the word bug in casual conversation and occasionally share a wall, but they belong to entirely different branches of the animal kingdom. Insects have 3 body segments, 6 legs, 1 pair of antennae, and in most species, wings. Spiders have 2 body segments, a fused head and thorax called the cephalothorax, plus an abdomen, 8 walking legs, no antennae whatsoever, and no wings. They're arachnids, members of the class Arachnida, and their closest living relatives aren't the flies or beetles they sometimes eat. Their relatives are scorpions, ticks, mites, and harvestmen. The chelicerates lineage, the ancient evolutionary branch that unites all of these, also includes horseshoe crabs. Despite the name, horseshoe crabs aren't true crabs. True crabs are crustaceans. A 2019 Harvard study placed horseshoe crabs within the arachnid radiation itself, part of the same evolutionary grouping as spiders and scorpions, rather than a separate chelicerate lineage. The animal harvested for its blue blood by pharmaceutical companies shares a deeper family history with the spider on your bathroom ceiling than it does with the crab on your plate. The practical consequence of the arachnid classification is that spiders can't be understood using insect biology as a template. Their sensory systems, their body plans, their evolutionary history, all of it developed on a completely separate track, under separate pressures, across a separate timescale, which is where the story actually starts. The Devonian period ran from roughly 419 to 359 million years ago. The National Park Service's geological record describes its significance plainly. Land plants were undergoing their first major diversification, the first forests were appearing, and the oldest spider fossils date to this time. That last detail tends to pass without registering what it means. To register it properly, you need to rebuild the Devonian world from the ground up. Start with what isn't there. There are no flowering plants. Angiosperms wouldn't appear until roughly 130 million years ago. No grasses, no fruit, no leaves in the broad, flat sense we recognize. No bees, no butterflies, no birds overhead, because birds hadn't evolved yet. Because the dinosaurs that would eventually give rise to birds hadn't evolved yet. The sky is empty. Literally empty. Of anything with feathers or scales or a beak. What's on the ground instead? Spore-bearing plants, low and primitive, rhiniophytes and lycophytes and liverworts spreading through wet ground near standing water. Early vascular plants were only beginning to develop the structures that would eventually become wood. The first proto-forests were tall by comparison to what came before, but they were strange, more like a densely packed collection of green vertical tubes than anything recognizable as a tree today. The air smelled different, tasted different. Oxygen levels were fluctuating through the Devonian, sometimes significantly higher than present. The humidity near waterways was high. There was no birdsong because there were no birds. No insect buzz at flowering plants because there were no flowers, and most winged insects hadn't developed wings yet. What sound was there would have been wind, water, and the movement of the arthropods that were colonizing this landscape for the first time in complex, predatory numbers. Into this world came the arachnids. The earliest unequivocal terrestrial scorpions appear in the fossil record at around 430 million years ago, in Silurian period rocks from the Pentland Hills in Scotland. The species is named Dolichophonus loudonensis. By the late Silurian, scorpions were established on land. Trigonotarbids, an extinct arachnid order regarded as among the closest relatives of spiders, with 8 walking legs and book lungs confirming fully terrestrial respiration, appear at the Rhynie Chert fossil site in Aberdeenshire, Scotland, preserved in exceptional detail at roughly 410 million years ago. The Rhynie Chert is a Lagerstätte, one of those rare geological formations where soft tissue is preserved in enough detail to see individual anatomy, entombed in ancient hot spring silica. The trigonotarbid preserved there, Paleocarinus, was a fully terrestrial predator moving through the earliest land plant ecosystems, hunting through the base of those primitive vertical plants, ambushing myriapods, early centipede relatives, and other arthropods that were also making their first attempts at surviving on dry land. These weren't insects. They weren't yet spiders. They were something older, operating the same basic chassis: 8 legs, book lungs, chelicerate mouthparts, in a world that was essentially inventing land predation as it went. A proto-spider called Attacopus fimbriunguis, found in Middle Devonian rock in Gilboa, New York, dates to approximately 386 million years ago. When first described in the late 1980s, it was classified as the oldest known spider. Later work revised that. Attacopus had silk-producing spigots, structures preceding true spinnerets, but lacked the abdominal tip spinnerets that formally defined the order Araneae. It now sits in an extinct precursor group called the Uraranida. Attacopus isn't quite a spider, but it produced silk, it hunted, and it's what a spider came from. True spiders, animals with functional abdominal tip spinnerets, appear in the late Carboniferous, around 315 million years ago. The numbers are large. They need a comparison to become visceral. Bees. The fossil and molecular record places their first divergence from sphexiform wasps in the later part of the early Cretaceous, roughly 125 million years ago. The oldest bee fossil found in mid-Cretaceous amber from Myanmar is approximately 100 million years old, carrying pollen on its body. The Hymenoptera order that bees belong to is older, reaching back to the Triassic, but bees, as a specific pollinating lineage, are a Cretaceous innovation. Butterflies appear in the fossil record in the Eocene epoch, roughly 40 to 50 million years ago. Lepidoptera as an order is older, with Jurassic period moths, but butterflies as the distinctive wing-scaled daytime flyers that define the word butterfly to most people are geologically recent. Do the arithmetic. Arachnid ancestors of spiders were established as land predators more than 300 million years before the first bee. The spider lineage predates butterflies by something closer to 370 million years. The ecosystem most people picture when they think of nature, flowers, pollinators, color, birdsong, is a recent arrangement. Spiders were hunting through a world without any of it, catching prey that had no wings and no flowers to visit, in a landscape without a single angiosperm, for hundreds of millions of years before that world assembled itself around them. A University of Florida molecular dating study estimated arachnid origins at roughly 400 to 450 million years ago, consistent with the fossil record and likely extending beyond it. The researchers, analyzing complete mitochondrial gene sequences from 25 invertebrate species, including 4 spiders, 3 scorpions, 10 ticks, and 4 mites, noted that the fossil record probably undercounts arachnid age because the bodies contain no bone and decompose entirely. Their conclusion? These invertebrates appeared far older than the physical fossils alone would suggest. Research on spider body plan stability indicates that the fundamental architecture, 2 segments, 8 legs, chelicerate mouthparts, silk production, has remained broadly conserved for something approaching 350 million years. What you are looking at when you look at a spider isn't a modern adaptation to modern conditions. It's a blueprint that was already working before the continents were arranged in anything like their current positions, before the Atlantic Ocean existed, before the first flower bloomed anywhere on Earth. That stability sets up the next question. What did 350 million years of refinement produce? The web isn't a passive trap. That assumption is the next thing to collapse. Imagine an orb weaver sitting at the hub of a completed web at 3 in the morning. No light whatsoever. The spider's image-forming vision, such as it is, registers nothing. But 8 legs are in contact with the radial threads simultaneously, and each leg is essentially a listening post. The web is under tension. The silk was placed under pre-stress during construction, and that tension is deliberate. It matters acoustically. A moth strikes the capture spiral on the outer edge. The impact compresses the web slightly in that sector, sends a wave along the radial thread it hit, and that wave propagates inward toward the hub. The radial threads in an orb web transmit frequencies ranging from roughly 1 to 10 kHz efficiently, according to laser vibrometry measurements of orb weaver silk. A typical insect impact produces vibration signals that cluster around 100 Hz. The spider at the hub, sitting in total darkness, receives those signals through mechanoreceptors embedded in the cuticle of each walking leg. The primary vibration-detecting structures are called lyriform organs, groups of slit sensilla arranged in harp-like parallel arrays. Slit sensilla are strain detectors. Each one is a narrow groove in the exoskeleton lined with sensory neurons that fire when the groove is deformed. Thousands are distributed across the body. The metatarsal lyriform organ on each leg, a compound structure at the tarsal metatarsal joint known in the research literature as HS10, is described by researchers as highly vibration sensitive. 8 legs, running this system simultaneously, give the spider a directional array covering all incoming signal paths at once. The spider turns toward the sector of maximum signal amplitude and begins moving outward along the radial thread. It doesn't see the moth, it doesn't smell it. It feels the frequency pattern in the silk, distinguishes it from wind-generated noise, which has a different amplitude and pattern signature across the radial threads, and navigates to the prey source on vibration information alone. In complete darkness. National Geographic described the web in a 2014 feature on the research as a stringed instrument, a spider's silk web functioning as more than a home. That framing isn't poetic embellishment. The silk can be tuned, the spider adjusts thread tension during construction, and the frequencies it carries are what tell the animal where to move and why. The exact localization computation hasn't been fully resolved in the research. The animal clearly compares signals arriving along different radial threads, reading amplitude, timing, and frequency, but the precise algorithm remains an open question. The outcome isn't in question. A spider crosses several centimeters of web in darkness and bites prey it can't see, guided entirely by what its legs felt in the silk. Research published in 2022 by a Cornell University team extended this picture further. A single strand of spider silk, they found, is thin and sensitive enough to detect the motion of vibrating air particles within a sound wave. The same physical mechanism has a trichobothrium hair, operating at web scale. An orb weaver's web, depending on geometry, can present a sound-sensitive surface area up to roughly 10,000 times larger than the spider's body. The researchers observed orb weavers changing posture, turning, crouching, flattening against the web in response to sounds delivered at a distance, confirming that the web mediates acoustic detection independently of direct body contact. The animal has functionally extended its sensory surface onto its architecture. What looks like decoration is an array. That web gets rebuilt regularly. Many orb weavers start from scratch every night. Before rebuilding, most of them eat the old one. The behavior looks strange from outside. From inside the biology, it's precise. Spider silk is composed of spidroin proteins, metabolically expensive amino acid chains that the body invested energy in synthesizing. Those proteins are hydrolyzable. When the spider ingests the old web, proteolytic digestive enzymes break the spidroin chains into their constituent amino acids, which re-enter the body's protein pool and become available for the next web's production. Research on orb weavers, particularly species in the genus Araneus, suggests that under appropriate conditions, roughly 90% of the protein investment in a web can be reclaimed this way before reconstruction begins. That figure is specific to particular species under particular conditions. It isn't a universal statement about all spiders, but in the species where it has been studied, the recycling efficiency is remarkable. 90% recovery. The raw material for tonight's web is substantially last night's web, digested, reprocessed, and extruded again through glands calibrated to the current situation. A manufacturer recovering 90% of its primary feedstock before producing the next unit is running a sophisticated materials loop. The spider at the center of the room that most people walk past is running a sophisticated materials loop. What gets rebuilt isn't a copy of what was torn down. Research on Nephila pilipes found that web architecture, silk tensile properties, and silk amino acid composition all adjust in response to the spider's recent nutritional history. A rebuilt web reflects current conditions, different thread spacing, different tension, different silk composition, implying that the animal isn't repeating a fixed template but dynamically updating its instrument. The ratio of MASP1 to MASP2 in the new silk shifts based on what the spider needs. More MASP1 for stiffness and strength when structural rigidity matters, more MASP2 for elasticity and toughness when extensibility is the priority. The body plan includes a feedback loop between diet, silk chemistry, and web architecture. That loop has been running, being refined, for longer than the Atlantic Ocean has existed. The sensory architecture doesn't stop at the web. Distributed across the exoskeleton of a spider, not concentrated in any recognizable organ, not arranged in anything resembling an ear, are long, fine hair structures called trichobothria. Picture them as extremely slender, almost transparent filaments anchored in tiny, flexible membrane sockets across the legs and body. In the wandering spider Cupienius salei, a species extensively studied in sensory biology, researchers have counted approximately 900 of these hairs across the body surface. Each stands clear of the surrounding cuticle, catching the motion of whatever air passes around it. When air moves, even the tiny displacement of air molecules within a passing sound wave, a hair deflects. The deflection might measure nanometers. The socket membrane at its base deforms under that pressure, stimulating sensory neurons. The signal moves into the nervous system. What trichobothria detect, technically, is near-field air particle velocity, the oscillating back and forth movement of air molecules within a sound wave at close range, rather than the far-field pressure waves that vertebrate cochleae convert into hearing. The distinction matters for precision. Trichobothria aren't ears. They are mechanoreceptors tuned to the physical medium that also carries sound. Research has measured their sensitivity across a range of roughly 10 to 950 Hz, a window that spans the wingbeat frequencies of many flying insects. In 2016, Paul Shamble and colleagues at Cornell University published findings in Current Biology demonstrating that the bold jumping spider, Phidippus audax, perceived airborne sounds at distances exceeding 2 meters, well beyond what near-field velocity detection alone would predict. The spiders responded to low-frequency tones at around 80 Hz by freezing, a standard anti-predator response that requires the animal to have registered the signal as a potential threat. The researchers also recorded electrical activity directly from neurons in the jumping spider's brain during acoustic stimulation, and confirmed that the signals were being processed at a neural level. The mechanism throughout was trichobothria, an animal with no ear structure, no tympanic membrane, no cochlea, no auditory canal of any kind, detecting and responding to airborne sound at more than 2 meters through hairs on its legs and body. Stack this against what the web already does. An orb weaver sits at the center of a silk structure that functions as an acoustic array covering thousands of times its own body area, detecting air particle motion within sound waves passing through it. The spider's own body carries approximately 900 hair mechanoreceptors sensitive to the same physical phenomenon at the near-field level. 2 overlapping layers of sound-adjacent sensing, neither of them an ear, working simultaneously, covering different spatial ranges. A 2024 study on the net-casting spider, Dinopis spinosa, found it could detect auditory stimuli from at least 2 meters at or above 60 decibels sound pressure level, corroborating that far-field sound detection through trichobothria isn't limited to jumping spiders. The predator that appeared to be a simple mechanical trap is running a multi-layer sensory system that researchers are still characterizing. The predator is also not limited to the menu most people assume. Spiders are generally described as obligate predators, animals that hunt and consume other animals exclusively. The record disagrees. Bagheera kiplingi, a jumping spider native to Central America, is formally classified as predominantly herbivorous. Its primary food source is Beltian food bodies, lipid and protein-rich leaf tips grown by acacia plants as rewards for their ant symbionts. Bagheera kiplingi bypasses the ants and takes the food bodies directly. Research confirmed this in peer-reviewed literature. It remains the only spider currently known to be primarily plant-eating. That is an outlier. But the broader pattern of dietary flexibility is documented more widely. Research published in the Journal of Arachnology recorded spider species consuming pollen trapped in their webs as a regular nutritional supplement. A study in Current Zoology, examining the cursorial spiders Carotus videus and Oxyopes javanus, demonstrated that sucrose feeding improved survivorship in animals deprived of insect prey. Research on crab spiders of the species Ebrectella tricuspidata found a measurable preference for honey solution over plain water. A formal study titled "Feeding on Nectar and Honeydew Sugars Improves Survivorship of Two Nocturnal Cursorial Spiders," published in the Journal of Arachnology, established that plant-origin carbohydrate sources extend survival through prey gaps. Fungus and fruit consumption by spiders and harvestmen has also been formally documented in the same journal. This flexibility isn't incidental. It's a survival feature built into a body plan that was always going to encounter environments where insect prey is scarce. Cave systems, building interiors, spaces where nothing flies and nothing crawls in for weeks. The spider in the empty cellar isn't slowly starving. It's finding something to eat. In the dust, in accumulated organic material, in what looks to any human observer like nothing at all. The architecture was built for exactly that situation. There is one type of spider that tends to shift the assumptions even for people who are certain they don't find spiders interesting. Jumping spiders, family Salticidae, have a visual system built around two large, forward-facing principal eyes that work optically like telephoto lenses. Each produces a high-resolution color image from a narrow field of view, roughly 1 to 2 degrees. The retinas of those principal eyes are layered in tiers, and the spider achieves depth perception not through muscular accommodation of the lens the way human eyes do, but through a shift in retinal position relative to the focal plane. The eyes themselves can rotate slightly within their sockets, tracking a moving object without the spider turning its body, which creates the characteristic impression of an animal that is watching you very specifically, from a face that isn't supposed to be capable of that kind of directed attention. Secondary lateral eyes, arranged to cover the wide field, detect movement and trigger the principal eyes to reorient. Research on visual acuity in salticids has placed them above mice and rats, and in some comparisons approaching pigeons and cats for image resolution relative to body size. The consequence of this hardware is observable behavior. A jumping spider approached by a moving finger tends to stop what it was doing, reorient both principal eyes toward the stimulus, and pause. Not for a fraction of a second, for a sustained period that registers as assessment. The animal is looking, and the looking is directional, focused, and followed by a decision: advance, retreat, or ignore. People who keep jumping spiders as pets report with notable consistency that the animals track them across a room, respond differently to different individuals, and sometimes react to a hand appearing through a window several centimeters away. These are informal observations. They describe behavior consistent with what the visual hardware is capable of, but they aren't controlled experiments, and they shouldn't be presented as proof of anything beyond what the hardware can support. The genus Portia takes this significantly further. Portia spiders are araneophagic jumping spiders. Their prey is other spiders, and they approach that prey through documented behaviors that have occupied researchers for decades. One is deceptive web plucking. Portia enters the web of a resident spider and plucks the threads with its legs and palps in rhythmic patterns that mimic the vibration signature of struggling prey or a courting male, manipulating the resident spider's orientation and response until the opportunity for an attack presents itself. The tactic varies between species and adjusts based on feedback. If a particular signal produces no response from the resident, Portia tries a different pattern. A second documented behavior involves planned detour routes. When a direct approach to prey is blocked or carries unacceptable risk, Portia selects long, indirect paths that require breaking visual contact with the target, maintaining a spatial representation of where the prey is relative to the route's endpoint, and arriving in striking position without a continuous sightline during the approach. A paper in Frontiers in Psychology, titled Arthropod Intelligence: The Case for Portia, documented pre-planned detours, expectancy violation responses when anticipated prey was absent, and novel problem-solving across multiple Portia species. The authors were careful with their language. They weren't claiming vertebrate-equivalent cognition, but the behaviors they documented don't fit a pure stimulus-response model. What this adds to the portrait is pressure on a category, not proof of a new one. An animal running deception through signal mimicry, navigating spatial detours that break sightlines, and responding to novel situations with something that observationally resembles decision-making, isn't accurately described by the phrase mindless bug. Whether something more precise than that phrase requires the word cognition is a question the research hasn't closed. The behavior is there in the literature. The framing sufficient to contain it isn't yet agreed. The predatory system has one more piece, and it's the strangest. Spiders don't chew. The chelicerae that grip and puncture prey aren't structured for grinding tissue. Instead, venom is injected through the chelicerae at contact, and that venom contains digestive enzymes. Those enzymes begin dismantling the prey's internal tissues immediately, before the spider has finished securing the capture, sometimes before the prey has stopped moving. The process produces a liquid, partially digested tissue drawn back through the chelicerae into the spider's digestive system. The cycle repeats until the prey is substantially liquefied and the extractable content is gone. What remains on the web is a dry, thin husk, the chitinous exterior emptied. Research on spider digestive chemistry has identified proteolytic enzymes as the primary class involved, protein-degrading compounds that break peptide bonds throughout the target's tissue. Some researchers working on spider enzyme profiles have observed that these proteases appear capable of solubilizing silk proteins as well, suggesting that the same enzyme system used to dissolve prey may also participate in recycling old webs. Digestion began outside the body, in a system that serves simultaneously as weapon, processing unit, and possibly materials recovery tool. The reason none of this feels threatening in any immediate practical sense is scale. Every system described here, the vibration hunting, the acoustic web, the trichobothria-mediated sound detection, the silk recycling loop, the external digestion, the cognitive pressure from Portia's detour planning, the diet flexibility, runs inside a body that, in the species most people encounter, measures less than a centimeter across. The engineering stayed small. That is the only reason the corner of the room feels safe. Put the full picture together, and the phrase, ordinary spider, stops functioning as a useful description. What's in the corner belongs to a lineage whose arachnid ancestors were among the first complex predators to work on dry land, hunting through ferns and mosses and enormous arthropod prey in the Devonian, more than 300 million years before the first bee, roughly 370 million years before the first butterfly, in a landscape without a single flowering plant, without color in the way we now mean it, without anything flying overhead that carried wings. The lineage that gave rise to true spiders had been refining this body plan across that entire span of time. Research suggests the fundamental architecture has been conserved for something approaching 350 million years. From that architecture come systems that still feel alien when examined closely. A silk that outperforms steel by weight, composed of spidroin proteins arranged in crystalline nanostructures and amorphous elastic regions. recovered and rebuilt at roughly 90% efficiency in some orb weavers before each new night's version is deployed, customized to current conditions. A web that functions as a multimodal sensory array, transmitting vibrational information across a frequency range of 1 to 10 kHz, allowing the animal to locate prey in complete darkness on signal alone. Approximately 900 trichobothria distributed across the body in some species, responding to air particle motion from 10 to 950 Hz, extending sound detection to distances of more than 2 meters without anything resembling an ear. A diet flexible enough to include pollen, plant sugars, nectar, and honey solution when insect prey is unavailable, keeping the animal alive in environments where nothing else survives for long. A digestive system that begins outside the body, inside the prey. And in jumping spiders, a visual system with resolution approaching some vertebrates, combined with documented behaviors, signal deception, spatial detour planning, apparent assessment of novel stimuli that resist easy categorization. All of that is running in a body smaller than your thumbnail. The demo with the steel wire is where most people's surprise ends. It shouldn't be. The silk is the clearest place to see that the usual assumptions don't hold. But once you look at the silk carefully, you look at the legs, then the web, then the time scale, then the sensory systems, then the diet, then the behavior, and each assumption fails in order. Not because spiders are extraordinary in some vague sense, but because the specific systems are specific in ways that don't match the casual category they've been filed under. Normal spider is no longer a usable phrase. There is just this animal, ancient, precise, running surplus engineering in a very small body, doing exactly what it has been doing since before the world outside your window existed in any recognizable form. Subscribe if you want more stories like this.